Hello, my name is Harrison Liu, and in this presentation I'll be talking about the PD pathway, including its involvement in T-cell suppression and tumors' ability to evade immunosurveillance. I will also be mentioning the new area of immune checkpoint blockades that involve the disruption of this pathway. PD-1 is a membrane protein that is present on the surface of T cells. It is specific for PD-L1 and PD-L2, which are both ligands for the PD-1 receptor. Upon binding to PD-L1, PD-1 activates and suppresses T-cell activation. This correlates to a lower level in cytokine release and T-cell proliferation. This is vital for self-tolerance, which is why we see PD-L1 expressed on antigen-presenting cells to prevent T-cell activation, leading to autoimmunity, as seen on the right of this slide. Certain tumors have developed the ability to express PD-L1. This allows them to exploit the PD pathway and suppress T-cell response. This diagram shows one way a tumor can express PD-L1, which binds to the PD-1 receptor on the tumor-infiltrating T-cell. This suppresses the T-cell's response to the tumor cell. PD-L1 positive tumors has been correlated to worse prognosis in cancer patients. This raises the question of how we can stop tumors from exploiting this pathway, which could lead to better immunoactivity against this tumor cell. If we can somehow inhibit the binding of PD-L1 on tumor cells to PD-1, we could stop the tumor's ability to suppress T cells and allow the T cell to recruit more lymphocytes to battle the tumor. This has led to the development of a relatively new treatment called immunocheckpoint blockade therapy. It utilizes monoclonal antibodies to prevent the binding of PD-L1 to PD-1. As shown in this diagram above, there are currently two main immunocheckpoint blockade therapies with PD pathway. One uses anti-PD-1 antibodies, which blocks the PD-1 receptors on the T cell. The other uses anti-PD-L1, which bind to the PD-L1 on the tumor cell surface. One such drug, called nivolumab, is an FDA-approved antibody that binds to PD-1 and blocks the tumor's ability to suppress the T cell. Although there has been a lot of success in drugs that disturb this PD pathway to treat cancer patients, there needs to be more research done on how this type of therapy will interact in different tumor microenvironments. For example, one research shows that tumor environments with the presence of Tregs can actually downregulate T cell activity when treated with checkpoint blockade therapy. That is all for my presentation, and thank you for listening.